Hello everyone, I'm very happy to attend the ninth Asia and Korea Conference. My name is Ziyu Deng. I am from Wuhan City, which is located in the south of China, and I am a doctoral student of the last year at Huazhong Agricultural University. My major is food science and technology. Now, as an exchange student, I am at Nanyang Technology University in Professor Cher's lab. Today, I am very glad to share my research with you. The title is Research on Plant-Based Material Structure Regulation as Delivery Vehicles for Bioactive Macromolecules. I would like to introduce the following aspects. First, research background. Lactose is the main nutrition source until weaning, accounting for around 2% to 8% milk weight. At the same time, lactose also existed in many foods, such like sugar, bread, fruit juices, and so on. It is digested in intestines by lactase. More than 70% of the world population has varying degrees of lactase deficiency. As shown in the table, different races have different dairy food cultures, which results in different lactase digestibility. As far as Asia is concerned, the prevalence of lactose intolerance is as high as 95%. Research predicts that in 2025, about 80% of the world population will suffer from varying degree of lactose intolerance. Therefore, lactose intolerance has already related to the nutritional and health problem of the world population. Lactose intolerance is mainly caused by the lack of lactase produced by our small intestine. After the lactose in food enters the small intestine, the lactose cannot be broken down into glucose and galactose. As shown in the picture on the right, after lactose enters the colon, it is fermented by bacteria to produce organic acid and gases. This process can cause bowel, abdominal pain, and loose bowel. So these symptoms are called lactose intolerance. Up to now, the current solution to solve lactose intolerance are mainly the three one. The first is to produce lactose-free products. This is no doubt that this math is an industrial solution. However, many lactose intolerant individuals are not adjusted to the taste of lactose-free milk, such as the increased sweetness. Also, recent studies have indicated that the lactose-free diet may lead to inadequate calcium and some other health problems. The second approach, enzyme supplement, is a promising approach which will not cause undesirable changes in food quality or nutritional profile. However, there are two problems of this approach. One, market research has found that lactase enzyme supplement, typically in form of tablet, calcium, pill, or drop, which can help promote lactose hydrolysis in lactose-containing food. Liquid lactase is suitable for infants who cannot swallow or chew. This liquid lactase contains almost 50% of glycerin, since glycerin can stabilize lactase under heat treatment. But the excessive intake of glycerin can cause some unhealth symptoms like dizziness, and glycerin is not a health choice for infants and young children. Secondly, the effect of lactase is related to the activity maintained in the human gastrointestinal tract, but which is easily destroyed by gastric acid when it is taken orally. Hence, oral administration of enzyme remains a challenging issue. The third approach study have showed that a variety of probiotics can break down lactose and relieve symptoms of lactose intolerance. So, probiotic supplements, which on the market are very common and can be used on the basis of diet. In general, the method of enzyme supplement is the most direct treatment for lactose intolerance and does not cause changes in food quality and nutritional status. But there are still many problems. So my research focuses on the two major problems of this method. I would like to prepare a new lactase preparation with good stability, high utilization rate, and high nutritional value. Next, I would like to report on the progress of the research. The research progress is divided into three parts. The first part is about oligosaccharides act as the high-efficiency stabilizer for lactase under heat treatment. The aim of this study is to replace the glycerin in the commercial lactase with more healthy oligosaccharides, which is useful in enzyme supplement for infants. And our experimental research show after heat treatment, the native enzyme retained only 20% activity, while the activity retention rate of lactase with oligosaccharides reached 82%. On the other hand, we also provide an insight into the mechanism by which sugars stable protein under heat stress. So, these are our work highlights. This research obtained may produce a guideline for a novel liquid lactase supplement and provide more information for application of liquid lactase in food industry. On the basis of the previous work, the next work is aimed to solve the problem of the low activity of lactase supplement in processing, storage, and gastrointestinal environment. The scientific inspiration for this part of the work comes from the pollen and the spores of plant organic in nature. Everything created by nature has its specific mission and therefore has different characteristics. There are many advantages of pollen. So, in my aim is to develop a novel pH-responsive carrier to regulate lactase release. Like SEC is extracted from natural plants, so that the lactase complex is absorbed into the SEC and then coating different substances, so it can be taken orally through the stomach. In summary, we need to the second part of my content. To study SEC from different sources, comparing their structural characteristics and comparing which source of them have the betting, loading, and sustained release effect. The research results show that sunflower pollen has the best loading effect. On the one hand, the pulse on SEC could provide a pathway for lactase to enter the microcapsules, but on the other hand, the exchange of the pulse will cause the leakage of enzymes in the environment of processing, transpilation, and the gastrointestinal tract. Therefore, the polarly coating technology is used to coat the pulse on SEC. So the next part of the work is to regulate the properties of the coating by regulating the interaction among the protein, polysaccharides, and some small molecules so as to construct and compare different release galactic models to achieve optimal results. So in this part, we try different binding models, such as physical binding, no-covenant binding, coordination binding, and covenant binding to compare to study the protective effect of different binding models on lactase. Long-covenant binding is protein and polysaccharide system, and coordination binding is polysaccharide and mantle ion system, and covenant binding is protein and polyphenols system. The work highlight is on the natural polymaterials based on the interaction between food compounds. We prepared a long-term and stable enzyme delivery system, which solved the problem of poor stability of lactase oral preparation and laid the foundation for the development of new products. I think it not only provide a new direction and idea for nutrient delivery research, but also provide theoretical guidance for in-depth study of component interaction and their regulation under complex food system. My paper at this stage as follows, and my report is over. Thank you for your listening.